All right, now we're going to talk about section 2 of chapter 3 on chemical reactions following energy in our first section. I started in with the last section going into section 2 here, but I thought it'd be too long for a brief video, so I want to keep this one not terribly long as well. That said, learning objectives you should be able to accomplish following this section: explain what occurs in a chemical reaction, distinguishing between reactants and products, describing the three classifications of chemical reactions, and distinguishing between catabolism or catabolism and anabolism. Also discussing the exchange that takes place in an oxidation reduction reaction, explaining ATP cycling, defining the chemical reaction rate, and explaining activation energy. So starting with metabolism. Metabolism is simply the sum total of all chemical reactions in the body, or in this case, for a more general definition, all biochemical reactions in living organisms. A chemical reaction occurs when chemical bonds and existing molecular structures are broken. New bonds are then formed. It's expressed as a chemical equation, right? You have reactants on one side and products on the right side, or as a result. So we have reactants are substances that are present prior to the start of the chemical reaction, written on the left side, and products, substances formed by the reaction, written on the right side of the equation over here. Okay. In a balanced equation, the number of elements are equal on both sides of the reaction. Chemical reactions are classified based on three criteria: changes in chemical structure, changes in chemical energy, and whether the reaction is irreversible or reversible. Okay. Classifying these changes in chemical structure includes decomposition reactions, synthesis reactions, and exchange reactions. So starting with the decomposition reaction, the initial large molecule is broken down into smaller structures. So AB will be broken down to A and B. An example would be a hydrolysis reaction, so water breaking apart the bond, right, of sucrose into glucose and fructose. And all decomposition reactions in the body are referred to as catabolism or catabolic reactions. So we have sucrose molecule here, through hydrolysis, water added to the equation here, so to speak, break it apart, that bond, and then you get glucose and fructose, two separate molecules. In a synthesis reaction, two or more structures combine together this time to form a larger structure. So A and B separately become AB. An example is dehydration synthesis reaction forming a dipeptide from two amino acids, right? An anabolism is the collective term for all synthesis reactions in the body. So you have two amino acids and then water basically playing a role as well, as it always will with the bonds here, in this case adding to the equation and then forming a bond between these two amino acids to form a dipeptide. Okay. And in exchange reaction, groups are exchanged between two chemical structures. It has both decomposition and synthesis components. It's the most prevalent in the human body. Okay, we have A and AB plus C, and that converts to the products of A plus BC, right? Slight change, but nevertheless, there's an exchange here with both decomposition and synthesis components. And the production of ATP in muscle tissue is a great example of that. We have creatine phosphate molecule here. We have A plus ADP, adenosine diphosphate, and then that produces just a creatine, because one of the phosphates here comes off the creatine phosphate and binds with ADP, so you get ATP, adenosine triphosphate, and creatine. Okay. In an oxidation reduction reaction or a redox reaction, there's an exchange reaction where electrons are moved from one chemical structure to another. Okay, we've been talking about this all along. A structure that loses an electron is oxidized, and during the oxidation process, so to speak, and then a structure that gains an electron is reduced during reduction. Reactions always occur together. Electrons may be moved alone or with a hydrogen ion, and nicotinamide adenine dinucleotide, or NAD+, is an example. So here we have an NAD+. And the energy-rich molecule glucose, C6H12O6, is oxidized and gives up two hydrogen atoms. Okay, and that actually then NAD+ is reduced, gaining both hydrogen ion and two electrons. So here you have an NAD+, the oxidized form of nicotinamide adenine dinucleotide, NAD+, and basically add in the two hydrogen atoms here, and we get a reduced form of nicotinamide adenine dinucleotide, NADH. Okay. And the classification of chemical reactions, we basically have exergonic and endergonic reactions. Exergonic reactions, reactants with more energy within their chemical bonds than products. Okay, energy is released with a net decrease in potential energy. Decomposition reactions are an example of exergonic reactions, basically catabolic type reactions. Okay, where things are essentially oxidized. And they have endergonic reactions, reactions with less energy within their chemical bonds than the products, energy supplied with a net increase in potential energy, like synthesis reactions, where basically we're talking about anabolic and reduction. Okay. So here we have in our exergonic reaction, we have glucose and oxygen, and then they basically here in the exergonic reaction as they come down over here, we're basically talking about them being oxidized and brought into carbon dioxide and water. And then we have our reactants here, amino acids, energy supplied in an endergonic reaction, becoming products of basically of protein. And the endergonic reaction, the anabolic response, synthesis, right? And reduction. And then we have ATP cycling. We have continuous formation and breakdown of ATP. Your body's always using and then reformulating ATP. If it doesn't do it, the cell dies. It's that simple. ATP is formed when energy is released in exergonic reactions. Okay, and basically fuel molecules from food are oxidized, right? And then the energy in the bonds is transferred to ADP and free phosphate to form ATP. And ATP is oxidized to aid endergonic reactions, where energy is released from ATP through hydrolysis to provide energy. Okay, and only a few seconds worth of ATP is present at a time. ATP is not stored in the cell for more than give you energy for more than 1 to 5 seconds, as we said before. Okay, ATP is going to come up a lot in future units here. So ATP cycling, here we have essentially starting with ATP. If we have ATP in the beginning, the triphosphate group with ribose and adenine, and we have ATP splitting, and energy is released. We have potential energy here, chemical energy, chemical bonds here, potential energy, and then they are basically energy is released, right? Kinetic energy is then formed with a release of the electrons here, so to speak, and we have basically inorganic phosphates released here, and then basically that now we have ADP and that inorganic phosphate. And then after you get, hopefully that inorganic phosphate is going to try to bind again with ADP someplace else, and energy is supplied through an endergonic reaction, the formation of ATP, and it cycles again and again. Okay. Classification of chemical reactions. Reactions are either irreversible or reversible. Generally speaking, chemical reactions are reversible, but there are some irreversible reactions potentially. From a general standpoint, reversible reactions are reversible with the standpoint that maybe within minutes, seconds, minutes, days, but there are some reactions that may take millions of years to reverse. Okay, so irreversible is where you have a net loss of reactants and a net gain in products. Whereas a reversible reaction, there's no net change in concentration of either reactants or the products. Okay. The reaction rate is a measure of how quickly a chemical reaction takes place. Okay? Activation energy, or EA, is the energy that's required to break existing chemical bonds. It's a primary factor determining the reaction rate. Right? Generally speaking, overcoming the activation energy in a lab, temperature is often increased, providing energy to break bonds, and the body usually using more catalysts called enzymes to increase increasing rates of reaction within the body. Okay. And of course, if temperature increases too much in a cell in general, like someone having a fever, in general, too much, the body gets too hot, exerting at a high rate during high heat, and basically someone gets above 105 degrees and more is about the temperature where proteins begin to denature and unravel. Essentially, protein begins to unravel, which means protein being part of every cell. That's not good news, right? So what did you learn? The differences between reactants and products in a chemical equation. Well, we start with the reactants, and the end result after basically the chemical change, after this transformation or conversion, we end up with the products on the other side. For a biochemical reaction that involves simple chemical structures bonded together into a more complex molecule, right? Choose the more accurate term in each pair that best describes this type of chemical reaction. So we're talking about bonding together to form a more complex molecule. That is synthesis. Okay, and with that synthesis, when we're building up together here, we're talking about endergonic, and of course that is also anabolic. They all go together. Okay. What molecule is formed from glucose oxidation and used as the energy currency for energy requiring processes within the cell? That's ATP. Okay. What occurs when the equilibrium is disturbed in reversible reactions by changes in reactants and products? Well, basically, there should be no net change in a reversible reaction, right? However, there's a net loss of reactants and a net gain in products in an irreversible reaction. Explain the effect a fever would have on chemical reaction rates within the body. What's the risk to protein structure with a high fever? Well, as we increase temperature, right, as we increase temperature, the risk of proteins denaturing or unraveling becomes higher risk. And when that happens, when protein unravels, it's going to basically all tissue that's related to cells, which protein part of every cell can denature and unravel, and cells can die, which is obviously not what we want, which is why high fever is not a good thing, right? Generally speaking, for a cell life, we're talking about a significantly high fever, okay? That said, that is section 3.2. We'll come back to enzymes in section 3.3 shortly.